Embryonic development involves a large number of protein-coding genes, which encode signals and receptors for signals, and transcription factors, and kinases, which all have to be expressed at precisely the right level, in precisely the right area, at precisely the right time for development to occur normally. It turns out in order to get this correct expression of the protein coding genes, there are a large number of DNA sections encoding microRNAs which regulate the expression of these genes. Of course, DNA does not only encode protein encoding genes. We know that while only 3% of the genome codes for the amino acid order of proteins, about half of the genome is transcribed, forming a transcriptome with many RNAs which are never converted into protein. These include microRNAs, which are important in the regulation of the mRNAs for genes, given that they can bind to mRNAs and then either cause their degradation or block their translation into protein. These microRNAs are clearly essential for development. One can observe this in mice which have been engineered to not express specific microRNAs. In other words, they are gene knockout mutations so that one can observe the effect of the gene under normal conditions by observing the phenotype when the microRNA is not expressed. And so diverse microRNAs cause diverse defects, and many are lethal. And thus, there are diverse microRNAs expressed during embryonic development which have specific roles. So, for example, the formation of the neural crest cells, which make so many important vertebrate tissues. This tissue is not only under the control of protein-coding genes, but also microRNAs. The differentiation of cell types, such as chondrocytes in cartilage, or the cells of the nervous system, or the cells of the heart, or pigment-producing cells in epithelia, these are not only encoded by protein encoding genes, but also regulated by microRNAs.